Good morning everyone. Today we will discuss tools involved in recombinant DNA technology. What is recombinant DNA technology? It is a technique in which we modify a genome of organism by inserting a gene of interest from another organism. Here I draw steps involved in recombinant DNA technology. Then we will discuss them step by step. Assume we have a genome of one organism and we want a gene of interest. So what we do? We will cut the gene of interest. So we will utilize our first tools called restriction enzyme. What is restriction enzyme? It is an enzyme that cut double stranded DNA molecules. Restriction enzyme will act on a specific location on the DNA molecule. We call this recognition sequences. Recognition sequences. Recognition sequences are palindromic in nature, so we can call palindromic sequences. Why we are calling them restriction enzyme? Because all restriction enzymes are isolated from bacterial cell and it is a defense mechanism of bacterial cell. How? When a pathogen, example virus will act on bacterial DNA, then bacterial cell produce recombinant DNA and it will cut the viral DNA into the fragments. So, here I am writing some example of Rico R1. These are the example of restriction enzymes like in second, etc. Now we will understand the nomenclature system of the restriction enzyme. See, the first letter is represent their genus name, and second and third letter represents species name, and the fourth letter represents a strain from where we isolate this restriction enzyme, and the last Roman number represents a sequence from which we first isolate this restriction enzyme. Now, we will discuss here the mechanism of action. Of these enzymes. Suppose we have E. coli restriction enzyme EcoR1 and it will recognize its recognition sequence. Here I am drawing this recognition sequence like G A T T C. And same here G A T T C, and we'll add bond between them. Now, why we are calling them palindromic sequences? When we read this direction to this direction, the sequence is GAATTC, and we read 5 dash to 3 dash direction, it is GAATTC. It is the same from where we read from where to here and here to here. When we read a word from both direction and it is the same, we call them palindromics. Now, how it will cut the double stranded DNA? It will recognize the sequence and cut the double stranded DNA by cutting the phosphodiester bond between adenine and guanine base pair, like here, and it will produce two sticky ends. One is it will produce those two same sticky ends, and this is our fragmented gene of interest. We call this a desired DNA. Now, we find our gene of interest and we have to introduce it to the cell. And what we do, we need a vector that can transport our gene of interest from outside to into the cells. Now we use a second tool called cloning vectors. What are the cloning vectors? First, we will define the cloning. What is the cloning? Production of large copy number of our desired DNA molecule into the cells called cloning. Like we have a DNA and it will replicate and will make two identical copies, and they will replicate and make four identical copies. These techniques we call cloning. Now, what is a restriction enzyme or cloning vector? Suppose this is our cloning vector. Specifically, cloning vectors are a plasmid of bacterial cells or yeast cell, and they have a double stranded DNA contains some features. Now, what are features of a cloning vector? They have a origin of replication. What is ORI? A conserved DNA sequence from where replication starts. If we have a origin of replication sequences, then autonomously replication will start. And second is multiple cloning sites. What is multiple cloning site? These sites are specifically for restriction enzymes. They have restriction enzyme sites from where restriction enzyme can act, like we have E. coli, EcoR1 sites, and we can have in second, etc. And what is the role of multiple cloning sites? Suppose we have a desired DNA and we want to insert our DNA into this vector for transporting into the cell. So we need some restriction enzyme site where restriction enzyme can act, and we can put our DNA into this. So the third is selectable marker. Selectable marker are specifically antibiotic resistance gene, like ampicillin, tetracycline. They are in restriction enzyme sites. So what will they do? They select separate the transformed and recombinant cells from non-recombinant and non-transformed cells. Fourth, low molecular weight. Why we need a low molecular weight cloning vector? Because if we have a low molecular weight cloning vector, we can generate large copy number of our desired DNA fragment. So, let's discuss how will we make a recombinant DNA by using our desired DNA fragment and this cloning vector. So, suppose we cut our desired DNA by E. coli R1. Same, if we cut our desired DNA by a restriction enzyme, the same restriction enzyme will be used for cut the restriction enzyme sites or cloning vector. Remember. Then we will cut our DNA by echo R1. Echo R1 site is there, so this site will be fragmented. And so see, we generate here two same sticky ends. And here E. coli can also generate same sticky ends and they will complementary to each other. So we can ligate themselves by the process called ligation. For the process of ligation, we will utilize an enzyme called ligase. The ligase enzyme will make the phosphodiester bond between the nucleotides. Now, what we did, we find our gene of interest by restriction enzymes. We ligate our gene of interest into cloning vector and we make a recombinant DNA. Now we have to transform this cloning vector into the host cell like E. coli for the production of the protein. How we will transfer our recombinant DNA into the host cell? Assume there are many methods. What is transformation? Transfer of recombinant DNA outside to inside the host cell is called transformation, and they utilize the steps like chemical precipitation method. In this method, we have ice chilled salt solution of CaCl2, calcium chloride, and we have host cell and our gene of recombinant DNA. So, we will transfer our incubated solution into the 42-degree heat shock for a brief time. Then we will transfer back into the ice chilled salt solution and it will precipitate the cells and facilitate the transformation of our recombinant DNA. How? Suppose if we transfer our heat chill to back into